So, in case of evaluating the Miller and Urey experiment, we have found that there are four important points. That first, I was talking about methane availability. So, at the time Miller and Urey performed this experiment, it was believed that they stimulated the early atmosphere accurately by including high level of methane, right? So it was like we are maintaining the early earth situation, right? So methane was enough. For energy source they have used electrical discharge, right? So electrical discharge was a source of energy instead of UV light. So this was the source. And presence of water is again a most important thing, because all the biological processes takes place in the presence of water. And if you remember, I have talked about primordial soup concept. So in the water environment, amino acid tends to remain as a monomer. Because of hydrolysis, the protein molecules break down, right? So there is no chance of protein formation, but amino acid will be there, because hydrolysis, what they do, they convert protein into amino acid. So that's the reason he has found the traces of amino acid in the liquid sample. Nucleotides. So first of all, Miller and Urey, they have not able to generate nucleotide, because nucleotides are produced by some chemically synthesized, using a different approach. So the approach was different here, which Miller and Urey have not able to explain, right? So these are the evaluation of his experiment. Let's go ahead with the topic. Let's talk about how the self-replicating cells are formed, right? So let's talk about cells as the smallest unit of self-sustaining life, right? So how the cells are formed, that's a very important question, right? Because in the early stage of earth, uh, the molecules, everything was going on, the formation process was going on. So first we have to understand the cell. This cell is first of all considered to be a smallest unit of life, right? Considered to be a smallest unit of life. So a cell have following common features. So let's talk about the features. What are the features? First features, you will find that they are covered by cell membrane. So let's say, if suppose this is a cell, right? This is a cell. You will find this is plasma membrane. Then there are some kind of a liquid inside, which is called cytoplasm. Then there is a genetic material, right? So this is DNA, right? Then there are some ribosomes molecules which performs protein synthesis, and these ribosomes are freely floating. These are the common things you will find in all cells, and of course there is a liquid inside the cell, and that liquid is called cytoplasm. Fine. So what is the next common feature? The next common feature of all the cells is they have a genetic information. You will won't get any cell without genetic information. So genetic information in DNA molecule, right? So these are, so the expression of DNA molecule is done by ribosome. So we can continue by saying which is expressed during protein synthesis, because ribosome performs protein synthesis. So these two things you will definitely get. So before going into the feature, what are the uh, defining features? Like if there is a cell, so life, how the life is defined using the term, that was the question. So life can be defined by using below features, right? Like when you say life is defined, there are certain features which is seen in all the life. First, what are the features? Metabolic reaction. This is very common. When we say metabolic reaction, example is respiration, right? Digestion, reproduction, right? Next is nutrition. Third is excretion. And the fourth, reproduction. So on this basis, life has been defined. So when we say reproduction, reproduction is to pass genetic information to the next generation. Information to offspring. Fine. And then of course later on organism will undergo climatic stress, right? And the good genes will pass on, or the genes which are not useful, they are going to removed out from the organism, right? Next fundamental features is responding to the surrounding, right? Like we can say responding to the external factor. So the ability to receive and respond, both. Respond to stimuli. Now from where? Maybe from the external, right? And internal environment, both. Next factor, growth. They must grow. So these are the 6 features of all the life. if something is living, you will find these 6 common features will be there in them. Now let's talk about virus. So virus are considered to be non-living. Considered to be non-living. Now when we say non-living, that means there are something which is missing, right? Though they do all these processes, what I mentioned above, they do. But there are something which is missing, right? So what is missing? So missing part is, first, they lack cell structure. If there is no cell structure, no cell organelles. Right? There are something which they carry on, like metabolic process. So I can write carry out metabolic reactions. Even though they are not a cell, they carry out metabolic reaction and, let's say, nutrition. Right? But can they replicate independently? Answer is no. So you can also mention that they are also unable to replicate independently. That means these viruses need something which helps the genetic information to get replicated, right? So practically they are not cell. We are not considering them into the cell category. Let's talk about how spontaneous origin of cells happen. So let's talk about explaining the spontaneous origin of cells. Fine. So first of all, we know from the cell theory that all cells come from pre-existing cells, right? That is a fundamental cell theory. So I can say the first point, that cells are considered to be complex structure, right? That they or that can only form from cell division, or you can say from the division of pre-existing cells, right? Maybe there are some cells they have divided, that's why a new cells is formed. Now the question is, how did the first cell come into existence, right? That is the question. So okay, we agree with the statement, but still question is, what is the question? Question is, how did first cell come into existence? This is the question. And we can also say that, how did the first cell come from existence if there were no pre-existing cells to divide, right? Came into existence if there were no pre-existing cells to divide? This was the question, right? We all agree. But we also know that, what we know? We know that all cells, what they have? They have partially permeable membrane, right? Then have genetic information, and these information can be passed on to the next, right? What next? Capable of doing metabolic process, can perform metabolic process, right? Like growth, maintenance, reproduction, right? So assuming that the first cell did not arrive on earth from somewhere else, they must be originated, right? So that means we have to come to a point where we can say that it's not coming from anywhere. Maybe the first cell has been originated. Now the question is how and from where. So we can say that that they must have originated from non-living compounds, right? So let me add this statement. So what we are assuming? So I am assuming, later on I will prove, assuming that the first cell, right, did not arrive on earth from somewhere else, right? Then what has happened? They must have originated from non-living compounds, right? And this non-living compounds have made that primordial atmosphere on earth, right? So we can say that made up the primordial atmosphere at that time. So these are our assumptions. So this would be required following step if we are assuming. So even though we need some steps. So what are the steps? Let me write. So this would have required the following steps. Now what are the steps? Let's talk about that. First is simple organic compounds. Simple organic compounds. Now these simple organic compounds need to be synthesized, right? When we say simple organic compounds, the meaning is um, carbohydrate, right? Like glucose molecule, the glucose molecule, amino acids, right? Methane gas, right? So all these simple organic compounds needed to be synthesized from inorganic molecule. This was the first. So a possible mechanism for this process was demonstrated by um, Urey and Miller experiment, right? So we can say how I can support this with the help of Miller and Urey experiment, right? Next, what next? Simple organic compound need to be assembled, right? So can I say assembly of simple organic compounds? And when they are assembling, what they are making? Polymers. Polymers molecule. Now you got the polymers. Now there are some polymers. When we say polymers, what are the examples? There are so many, like polysaccharide, polypeptide, right? Polynucleotide. Right? So you can see all poly words are mentioned. That means they are the big molecule, right? They are complex, and they are made from simple. So that means among all these simple or assembly of the simple or organic compounds, which we are calling as a polymer, they must have self-replicating property. So right, so that means some polymers needed to develop the ability to self-replicate. So that let's talk about the genetic information or genetic material. These molecules are self-replicating, and then after that, membrane formation. So there are some polymers have made the membrane, right? So compartments, right? So we can say membranes needed to uh, to surround the polymers, because these polymers need a very optimum conditions to work, right? And when we are creating a surrounding membrane, that means what you are doing, you are creating compartmentalization or creating compartments where these compartments are made within the cell, or you can say with an internal chemistry different from surrounding. So these molecules should work independently without any hindrance of others. These molecules are get compartmentalized. So what we have found, we have found that there are some molecules which are self-replicating. So we have discussed about self-replicating molecules earlier also. Let me go up to show where we have discussed. Yeah, here it is. So you can see these are CO2, H2O, H2, CO2, NH3. These are organic. I mean, atmospheric conditions which have these gases. These gases are inorganic, and due to UV radiations, the some molecule starts making polymers. Right, amino acids has formed protein by polymerization. Simple sugar get polymerized to form carbohydrate. Right, nucleotides get polymerized to form polynucleotides. So this process was continuously going on, and a self-replicating molecule was also formed, and then later on the compartmentalization also begins, where some molecules need a separate conditions to work on. So that is also cell's responsibility to maintain, right? So what we have realized that there are DNA molecules which are self-replicating. Let's say this is a mother DNA, right? Or let's say old DNA. So these mother DNA, how they are making two? They are separated into, let's say, two DNA molecule, and a new DNA strand has been formed, right? So these are daughter DNA. So like this, self-replicating molecules are formed. So some polymer, this is called DNA replication. One DNA makes one more copy of the DNA. So, and then you will also get that some of the cells have membrane, right? How membranes are formed? Maybe in the topic of cell membrane you have understood the process, how plasma membrane consists of. This is phospholipid layer. I am creating a phospholipid membrane. These are the head parts. We know each phospholipid molecules have one head and two tails. So I'm just creating number, doesn't matter, right? So these are highly big. So you can see how I'm making. In the same way you can also make. These notes will help you to save your time in preparing the paper or preparing for the board examination or IB examination. So what we have done, let's say the DNA molecule, right? So these DNA molecule must be stored somewhere in a very defined manners, right? So this is called membrane formation or membrane uh, form around the polymers, right? This is just an example, and this membrane is made up of phospholipid, right? So consists of phospholipid, and each phospholipid has what? There's a head part and there is a tail part, right? So something like this. This is head, this is tail. Now if we'll talk about protocells, means the first cell, and the theory behind it, you will find that yes, whatever we are discussing is actually possible, right? So let's talk about protocell. Protocell, that means first theory. Fine. So in like a cell theory that is now highly um, described and most uh, accepted form, this is protocell theory, where we will talk about what are the points are there in the protocells theory, which was the first theory given by scientists. So this theory proposed, what they proposed, they proposes that a cell like compartment right? There are some and they are capable of some basic metabolic functions. Compartment capable of some basic metabolic function, and this has arose spontaneously, right? We don't know. And and the cells which are these compartments which are formed here, cell like compartments, we are calling them as protocells. Fine. Initially, yes, there was no genetic material, so let me write that. Initially, they would have lacked genetic material, but they were able to grow, right? So I can also mention that, but they would have been able to grow as well as divide. So that self-replicating molecule was not there, but cell was sufficient to divide. This was a very important, and the divided cells into daughter cells. So some daughter cells are produced. Eventually, these protocells would have acquired genetic materials, right? Maybe later on, not initially, eventually. These protocells would have acquired genetic material, because the process, because Urey and Miller also, they were not able to explain how genetic information is formed, right? Like generally RNA was formed as they evolve, right? So these are the protocell theory, and the protocells theory pretty much discuss about how the cells was there, what are the molecules are there, how they were dividing or not, do they have genetic material or not. So these are some important point. If the question asks to discuss about protocells, these points can be written. There is also a first theory on genes. Let's talk about that. Most important. Gene first theory. So in case of gene first theory, these theories state with the spontaneous development. So this theory talks about spontaneous development of a nucleic acid, and that has ability to replicate also, right? That had the ability to replicate itself. So they were dividing also, and evolution by natural selection would have resulted into genetic variants, right? Like sometimes we say this variant is not good, this variation is not good, this variation is good. So variation is the difference among the organisms, whatever differences you are seeing. So evolution by natural selection would have developed or result, right, resulted in genetic variants, right, and that could have developed a cell membrane and the other things. That was the theory, right? So let me complete the statement. Genetic variants that could have developed a cell membrane and basic metabolic processes, right? In the same way, there is also a metabolic first theory or metabolism first theory. So how the protocells theory, genes first theory, in the same way metabolic first theory was also formed. Now this metabolic first theory says that yes, life originates as a system of complete reaction, like something has formed with all the processes. When we say processes, that means everything is being self-replicating, processes are going simultaneously, reproduction is going on, nutrition is going on, all the things are going simultaneously. When we say metabolism, the meaning is activity of the cell. When we say activity, maybe constructive activity, right, or maybe destructive activity. When we say constructive, this is anabolism, and when we say destructive, we talk about catabolism, right? So what this theory says, this theory proposed that life originates, uh, theory proposed that life originates or originated as a system of chemical reaction, right, and these chemical reactions capable of sustaining life. Or sustaining itself, right? So they are sustaining life, they are sustaining the molecule itself, and this system would eventually evolve to form a cell. So it's a gradual process, not in one day. So we can say that the system would eventually evolve to form cell. And when the cell is formed, and genetic material, both, because cell is formed, that means genetic material is there, because who will give the instructions, right? This theory is actually favored by many scientists as a most life processes essential for existence of cells required energy, and that energy is coming from metabolic reaction. This was very clear. So scientists were self-aware, right? So there is no false commitment or a kind of false judgment, right? When you will talk about how these vesicles are formed inside the cells, you will realize that yes, this is practically possible. So let's discuss about formation of vesicles. right? When we say vesicles, we are talking about the small vesicles, how the membranes are formed, like spontaneous formation of vesicles. Let's talk about that. The spontaneous formation of vesicles, right? So membrane, first of all, membrane play an a very important role in cells because they separate the genetic materials and other biochemical processes inside the cell from outside, and this what we call them compartmentalization, right? So there is a process of compartmentalization, and this compartmentalization is actually separating, like let's say when you do something, you generally want to get uh, to not get disturbed, we want to get compartmentalized in one room or a kind of one place where you can do your task. So membrane has helped in the formation of that compartmentalization, right? So we can say that membrane play an important role in cells because they separate the genetic material, right? And when they're separating genetic material, what else? And biochemical process. Where? Where they are separating? Inside the cell. And from where they are separating? From outside. From outside surrounding. Right? So that means membrane formation would have been a very important role in the origin of life. Right? So if we'll talk about the membrane, let's talk about the membrane. So you will find that membrane is, when we say membrane, what is there? So membrane means it is made up of fatty acid and fatty acids and phospho phosphate molecule, or better we can write one word, phospholipid, right? So first is phospholipid, which is a major component, and these phospholipids are amphipathic molecule. When we say amphipathic molecule, because they have both hydrophobic and hydrophilic part, right? Hydrophobic, hydrophilic, both are there. And fatty acids are, I mean, if you see the phospholipid, you'll find lots of lipids molecules are there. So you can see something like this. This is head, this is tail. Tail is made up of fatty acids, right? This is made up of phosphate molecule, right? There is so this particular head part is polar molecule. They are non-polar. When we say polar, means they are hydrophilic, right? They can mix with water. Non-polar means hydrophobic. They can't mix. and in such conditions the molecules will be amphipathic, right? So when a few lipids molecules placed in water, what we see that they form a kind of monolayer on the surface. The polar parts of the lipid will be in the water, while the non-polar part will be outside, or I mean not outside, inside, like this. So right. So head part is outside, tail part is inside, right? So outer part is hydrophilic and the core part is hydrophobic. Why hydrophobic? Because all the lipids part are inside. Tail is the phospholipid layer. So imagine if there are many uh, phospholipid molecules are like this, they can form bilayer. Bilayer is more stronger, right? So if lipid molecules are added, they can form bilayer. When we say bilayer, how they look like? And let me draw. This is bilayer. Right? So you can see this is phospholipid bilayer. So these monolayer has formed bilayer. So that was now possible. I mean, we can practically do it also. So you can say that if more lipid molecules are added, then what will happen? You can say they form bilayers. And when bilayers are formed, and what you will find in the bilayer? Polar facing outward, right? With the polar part or polar part facing outward, outward the water environment, because they can mix environment, while non-polar they are inside, while non-polar will points towards each other, right? So this is how bilayer has been formed. And small vesicles, you can say, so I can say that these bilayers will spontaneously form microspheres, or you can say small vesicles, and this could possibly have formed a membrane later on, right? And this is how early years membrane were formed, right? So I can put a point. Later membrane was formed, which have made which has helped actually to form many things, right? So yes, practically it was possible. So these early membranes have one uh, motto. What is the motto of these membranes? The motto was to separate the internal things from outside, right? When you have something, sometimes we don't share with others, right? Like if you want to study, what you do? You be in one room and you study without getting disturbed, something like that. So we can say that these early membranes would have separated the internal chemistry of the cell from others, right? So it is theorized that the fatty acid could have combined with glycerol. So how the membrane was formed, let's talk about that. I am seeing phospholipid everywhere, but how phospholipids are formed, let's see the idea. So what is the theorized form? So theorized form is that fatty acid, right, combined with glycerol. How they combined? By condensation. And what they have formed? They form triglycerides, right? Like let's say there is glycerol molecule and one fatty acid tail, right? Or if there is glycerol molecule and two fatty acid tail, or there is one glycerol molecule and three fatty acid tails. This is called, if the three tails are there, we call them triglycerides, right? If two tails are there, we call them diglycerides, and this is called monoglyceride. So yes, practically it is possible that glycerol and lipid can form. Now the question is how triglycerides was used. Finally, these triglycerides has formed. They get triglycerides get phosphorylated. Phosphorylated means addition of phosphate. Phosphorylation. And what they form? They form phospholipid, right? And now you will realize that eukaryotic cells evolve to contain multiple internal compartments because there are so many phospholipids, so many phospholipid molecules forming a membrane, and which allowed further cell division, right? So in this way the molecule was produced. But when we talk about the genetic information, let's talk about that. RNA as a presumed first genetic material. Let's talk about that. How you can say. So When the early years was formed, we have found that the following had two things as formed in the early years. I mean, in the early life. So, what was formed? Self replicating molecule. Right? This was formed. We have seen so far. And second, we have seen catalyzed chemical reaction. Right? And one more thing we have seen, compartmentalization. Right? This was also there. So in the modern cells, these functions actually are carried out by DNA and enzymes, right? But neither of these have been present in the prebiotic world. So scientists believe like these things. Now what we say in can I say that uh, in modern cell or recent cell, these functions are carried out by DNA and enzymes, right? And but now scientists believe that RNA may have performed both these functions in the beginning. That's why the RNA concept has come up. So we can say scientists believe that RNA may have performed both of these functions. Functions where in early cell means function of being a genetic material and catalyzing the process. Since it can store genetic information as well have enzymatic property, that's why RNA world hypothesis has come, right? So we can say that since it can store genetic material or genetic information as well as have enzymatic properties, this is known as RNA world, right? RNA world hypothesis, right? So as the life evolved, DNA took over the role of genetic molecules while proteins becomes catalyst, right? So there are some division of labor also happened here. So you can say that later as life evolved, DNA took over the role of, let's say, genetic store molecule, genetic storage molecule, and protein. What they becomes? Biological molecule, or you can say biological catalyst, right? So they are catalyzing the metabolic processes. So as we are realizing that yes, RNA have done a very tremendous or very um, patent work in the beginning. So there are some properties of RNA that provide evidences that what are the evidences? So let me write the next. properties of RNA that provide evidence for this. What are the evidences? First, RNA can assemble uh, spontaneously from nucleotide, right? Second, RNA is self-replicating. Next, control rate of reaction. And the modern, we also call them ribozyme, if you remember. Ribozymes, these are the molecules which performs enzyme role, and they are actually RNA molecules. Now, evidence that RNA may have been evolved before DNA. How you can prove that? Sometimes they ask in 1 mark or 2 mark. So, evidence that RNA may have been around before DNA includes, how you can say, first, ribose, right? Ribose can be formed from methanol. So one of the main product of the Miller and the Urey experiment that has triggered everyone in the beginning. Though later on now we know that ribose is a part of nucleotide, right? So either it's a DNA or RNA, ribose is there. So ribose can be formed from where? From methanol, right? Next, deoxyribose in DNA, right, is produced from ribose. So you can also say that yes, ribose has formed first. So ribozymes are able to join amino acid together to form protein, forms an RNA template, right? So that is also main evidence. So ribozymes able to join acid together. When we say acids, we talk about amino acid, and what they are forming together, they forms protein, and this protein forms from an RNA template. So things are very uh, organized and systematically uh, proven, right?